Signal processing techniques for nanopore sequencing. I'm Venkata Raja Gosula, and this is the topic of my presentation. First, I'll give you a little background, talk about the ideal model, real model, proposed study, and expected results. Nanopore sequencing is a second generation sequencing technique. Oxford Nanopore and Genia are the companies I know of that are doing this. It's rolling out, but it's not commercial successfully yet. It does not require a PCR step. It uses a single molecule. The technology basically you separate the two strands of the DNA, send a single strand through the nanopore, and then the electrical signal relates to the base going through a nanopore at that time. It has high error rates, but it can be improved with signal processing. Okay, here's a graph of the nanopore, and as you can see, the DNA gets unzipped and sent through the nanopore, and a flow of ions through the pore creates a current, and then as the base goes through, it blocks that current, and that's what's detected at the adapter molecule at the base of the nanopore. Ideally, each base would produce a very good signature, as I'm showing here, ACGT. There'll be no variability between the nanopores or reads, no temperature variation, repeatable ideal pulse, fixed distance between the pulses, and fixed separation between the peaks of the different bases. So the algorithm to read out the bases would be very simple. Wait for the first pulse, wait for a half pulse to center the sampling, and then keep on sampling based on the thresholds, and keep going until you read that there's no current different than the baseline. In the real world, here's some data from Oxford Nanopore. There's a distribution on the residual current. Luckily, it's not that overlapping, so that's why we're able to do some reads. There's big variability between nanopores and reads, and there's temperature variation and environmental variation. The pulses don't look all the same, and there's variable distance between the pulses. And of course, there's a distribution of peaks per each base. There are several nanopore types. There's biological versus solid state, SNP versus threaded, and the speed through a nanopore is rate controlled because the base speed of 1 microsecond per base is you can't recover anything. And there's some research in 100 microsecond per base, but whatever Oxford is working on right now commercially would be 1 millisecond per base, and that's the only way they can recover data. So different types lead to different characteristics and error profiles, and therefore different signal processing recovery mechanisms should be used. So I would like to study, starting with the ideal model, adding the disturbances such as amplitude distribution, pulse shape, variation of the timing between the bases, and some noise, and then implement signal processing algorithms on top of this, and to recover the base information and understand which variables affect the performance. Another thing we could do is increase the speed of the bases as they go through the nanopore. Now the base information is blurred, but we can model that as a Markov process and implement Viterbi decoding to extract the bases. And then again the same thing, recover base information, see what variables affect the performance. Furthermore, we could add some DNA intrusive techniques, such as add known training signals in front, and that way we could use those training symbols to do automatic gain control for threshold setting, and also figure out the frequency of the bases as they go through to extract the symbol timing. We should also be interested in adding coverage to the nanopore sequencing because now it's just single molecule, and understand how much coverage will be needed and how does the performance improve. So the expected results would be improved nanopore performance with algorithm, signal processing techniques, and improved performance such as reduced error rates and higher throughput.